I'm Sean Smukler, an associate professor in the Faculty of Land and Food Systems and the director of the Center for Sustainable Food Systems at UBC Farm. We're here at UBC Farm on the unceded and ancestral territory of the Musqueam. The UBC Farm is a 24-hectare academic site where we have research, education, and community engagement all at the same location. It's the site of one of our mother farm trials for the organic science cluster. So our project is really about identifying practices that organic farmers can adopt to increase their climate resilience while also reducing their impact on the climate. What we're trying to do is also incorporate some indication of what the greenhouse gas impacts of their choices are and enable farmers to clearly understand how the choices they make relate to the climate.
We're definitely not seeing any emissions so far. I've had this running for quite some time now.
Yeah? How's the battery on these guys?
They last me the whole entire measurement, which is 2.5 hours right now.
So one of the indicators of environmental sustainability that we're really interested in this project is the impact on the climate. We have 8 different treatments in this experiment. So we're measuring nitrous oxide, carbon dioxide, and methane, the 3 most important greenhouse gases coming from agriculture. And the idea is to, over time, be able to quantify those emissions while at the same time measure changes in soil carbon to get an understanding of the net impact on the climate. So what we try to do is have a subset of the treatments that we've got out here deployed on what we're calling daughter or child farm sites. So these are collaborating farmers across the province, and we've identified 3 distinct regions, Vancouver Island, the Lower Mainland, and the Kootenays, that would represent the range of soils and climatic conditions that we would see across the province. By utilizing these collaborating farms, we're able to capture some of the variation that we might expect from these types of management approaches. And through the combination of the parent sites and the child sites, be able to communicate what are likely to be the results of these different approaches.
This is what we're calling an auto chamber. And the reason why we call it an auto chamber is because it automatically opens and closes when we need to take the measurement from these areas. It measures the greenhouse gases, temperature, and moisture. So this is done all automatically 24/7, 7 days a week. All of these plots are ever so slightly different. There is either 2 cover crops, 1 cover crop species, and then we do multiplying of them to the fertilizer treatments we have, which is lots of compost, not a lot of compost, and compost targeting the particular plant requirements for that particular season. And so you'll notice there's some clovers here and there's some rye here. So this plot is what we call a 2-species cover crop mix. So when we start growing beets, when we start growing cauliflower and all that good stuff, after harvest, we will take a sample of it and try to figure out how much yield is coming off of these plants.
So we are trying different practices in side-by-side experiments. We are collecting different indicators of performance of the soil and of the crop. And then we're doing an economic analysis. We recognize both the environmental and economic outcomes of any particular practice, so farmers have a full understanding of the choices that they're making.
These are what we call ion exchange resin probes. So they're the things that we didn't get to see in the field, but we get to see out here in the lab. Also, another good name to call it is a plant root simulator. They essentially simulate the root by picking up whatever the root may pick up in plastic form. What we do with them is essentially insert them into the soil, let them sit for about a week or 2, take them out, extract them, and look at how much is in these probes.
A lot of very simple soil tests can give you total nitrogen, but that's not a very helpful approximation because the difference between nitrate and ammonium, which are plant available forms of nitrogen, change all the time. And so we want to know more specifically what the plant can actually use because there's other types of nitrogen in the soil that the plant can't uptake. I think that data can be really helpful for researchers and farmers to understand how their plants are doing.
We're partnering with a number of different organizations, including the BC Ministry of Agriculture, the Delta Farmland and Wildlife Trust, the BC Agricultural Climate Action Research Network.
AgriScience is going to be working with Professor Smukler to help with organising field days, so that's showcasing the on-farm research, as well as workshops which will be talking a bit about the research outcomes. And finally, we'll be creating plain language research briefs to help producers and other non-technical audiences understand the research.
The important components of this project is to ensure that farmers are understanding how these practices perform under different conditions.
There's a lot of great on-farm research happening related to climate. However, we know that when it comes to climate change, this research can't be done in a vacuum. So it's really important to be sharing the research outcomes with people in the sector. So AgriScience is working with the OSC4 project to help mobilize that research to make sure many stakeholders know what's going on and can really use the results.
Farmers certainly are keenly aware of the impacts that climate change is having on them. In terms of their awareness of what they are contributing in terms of the changing climate is something that we would like to try to address.